Continue our discussion on cancer and variants. And yes, so if we're looking here, as you see in this slide, we are looking at the stages of the central dogma, right, that we discussed before. In other words, we're looking at how the mutations that takes place from the transcription would affect translation, eventually the protein function. Cancer in general, a disease that largely stems from genomic alterations, eventually causing what some issues or uncontrolled cell proliferation. This growth, uncontrolled growth of the number of cells, where specifically these cells have been some issues in the DNA. So somatic DNA mutations, like for example, in this case, we're looking at adenine being mutated to thymine in the DNA sequence. Now, this type of mutation, we discussed before different types of mutations. This is what we can refer to as the point mutation. Now, what we can see that this kind of mutation may impact later what? The amino acids. So in this case, we're looking at change from aspartic acid, ASP, to valine in a protein. So that A to T mutation would lead to this change. And so, as we know, each amino acid is made of 3 nucleotides in a protein. It's coded by these 3 nucleotides that we called a codon when we discussed before, the DNA, RNA, and protein. So the mutation in one of these nucleotides, which are part of that codon, which is part of the representing eventually the amino acid and being part of the whole protein sequence, can affect the protein function. So looking at this, specifically, can we face a dysfunctional protein, like take this example, the change from aspartic acid to valine. The aspartic acid is a negatively charged polar amino acid, whereas valine is a hydrophobic non-polar amino acid. So this kind of change from polar to non-polar amino acid can significantly alter the structure and function of the protein. This guy gives you an example of what we're looking at when it comes to issues of these variants, and eventually having a dysfunctional protein would mean we are having maybe loss of function. Maybe the protein acquired the new harmful function, so that's what we call gain of function, and or the altered protein may interfere with the function of normal proteins. So this is what we refer to as the dominant negative effect. So what we see here in this slide is, sorry, we're trying to explore some of the types of these mutations as we go through in the coming slides or variants. So this is a reciprocal translocation. So it's not just a normal translocation. This is a type of chromosomal abnormality where 2 chromosomes, as we see here, exchange the segments with each other. So we have this normal chromosome, and this is another chromosome. This is chromosome 9. This is chromosome 22. We know it's shorter. Remember the karyotype that we looked at before. So if we have this exchange, so we take this piece here, this part, okay, map it to here, and then this part would move to here. That is the exchange. Not only one of them, but both, okay? That's why it's called reciprocal translocation. What we're going to end up with is the change that we realize in chromosome 9 with this translocation, and what we see now, which used to be a normal chromosome, now is not anymore. This is what we refer to as Philadelphia chromosome. And Philadelphia chromosome is the chromosome resulted now because the reciprocal translocation created what is called the fusion gene. Okay, so this is now, this translocation ended up affecting the gene sequence, creating a hybrid gene formed from two previously separate genes. So now we have a hybrid gene. Like in this case, we're referring to this gene as BCR-ABL fusion gene. This can lead to production of abnormal protein and can eventually contribute to the disease development. So Philadelphia chromosome is one good example for this story. In chronic myeloid leukemia, and it results from this reciprocal translocation between chromosome 9 and chromosome 22. Okay. These kinds of reciprocal translocation are detected through some genetic tests, such that karyotyping, that we know this is a way to visualize the chromosome. What we see here, for example, is this kind of visualization for the karyotype. We're trying to visualize the chromosomes under a microscope, and we're trying to see use some advanced technologies like fluorescence and cytohybridization to be able to see the overall changes, like in this fluorescence and cytohybridization, or FISH for short, which is a molecular cytogenic technique. It uses fluorescence probes that binds only in particular parts of a nucleic acid. So what we see here in this figure, using this FISH technology, is there is this rearrangement. So the chromosomes are seen in blue. The chromosome that is labeled with green and red spots, and this area here at the upper left side, is the one where the rearrangement is present. So tagging using kind of tracing some of the genes and seeing what's happening is one way to try to follow up with if we are having these kinds of translocations or so. We'll see later on that next generation sequencing can also be used, especially when we're supplied with the information of paired end reads. So in this case, again, looking at this chronic myeloid leukemia, which is affecting the bone marrow, this is a severe kind of cancer, and if something is affecting the bone marrow, it's directly related to the immune system, and I think you would get it. It's white blood cells. Okay, so this disease is characterized by the overproduction of white blood cells, the abnormal white blood cells. Okay, these these are kind of immature white blood cells. Okay, the healthy, the normal white blood cells are playing very important role in fighting infections and and building or helping the immune system. And now this kind of disease is breaking this whole thing. This is a very tough type of disease. From my reading, it's very difficult to detect at early stages, okay, as compared to other types of cancers. So usually this is really a late advanced stage when they know about it. So these plots here that we see are showing summary of variants detecting among a cohort of cancer samples. So we can see they are categorized by source tissue or variant types, and we also see the patient samples from that cohort. So the idea here is that not all genes are equally likely to drive or be promoters of cancers. So this is important as we are trying to detect specific types of cancers. This is why I've referred, maybe I mentioned this before, there is this now protocol for breast cancer. So once they know it's there, they do genetic testing early in the treatment process. With genetic testing, they are aiming, as far to the best of my knowledge, and as far as being exposed to the story of one patient, roughly 18 genes. So of course it depends on the type of the cancer. So they're looking at these 18 genes, trying to see which kind of variants are there, and then do we have a mutation? If there are any variants, substitutions, insertions, deletions, what kind of rearrangements, what kind of mutations are we having there? Okay, and so here you can see the number or percentage of patients with that specific mutation, and within that bar we see the distribution based on the bar components here are colored according to the type of variants detected in that specific gene. Yeah, so here we have also the sources of tissues related to that. Allele frequency that we are going to speak about here, related to what we have discussed before in terms of the IGV viewer. It's not the traditional or the typical concept that we have visited in the beginning of the course. There we were looking more at the allele when allele changes in a population. So to get the allele frequency there, we have to look at the parents, maybe the person, or other samples, reference samples, not necessarily the parents, and so on and so forth, and try to derive the percentage. But here, for a variant allele frequency, we are looking actually at counting the number of reads over the total number of reads. So after we see this alignment and we see this coloring, which is indicating some types of changes or variants, what we can do is, in this case, if we zoom in, we can see that this is originally C, okay, and this is originally an A, and we know that the orange color, or this color from the reference color set, so you can see it here, is meaning guanine, okay? So in other words, this is an A substituted by G. It's originally A, and now what we see is orange color. And here, this is, well, this is, I guess, yep, C. Okay, so this is cytosine, but now flipped into guanine. So if I want to measure or know the variant allele frequency here, I'm going to count all the reads, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, you'll get about 23 over the total number of reads. We get 38%. While compared to A to G, you see that all reads are having that. So it's 1, It has the result of 1 or 100%. Okay. So these are kind of measures to indicate and define the variant allele frequency. So yeah, you can hover here and then get these numbers from the IGV viewer, which is really nice, and it's easily going to indicate for us what's happening with this alignment. Remember from before that there are different kinds of mutations as we can categorize by the observed effect. So there might be frame shift mutations. We're looking at deletion of some nucleotides or insertion of some nucleotides. There are point mutations, so it's single mutated nucleotide. Now, sometimes this might be silent, meaning you have a mutation here, but we don't see a recognized phenotype there. And sometimes could be related to some impact on the function. Okay? It could lead to some function change. So in this case, we're looking at missense, which is affecting the function of that protein that will be coded. So remember the example about, you know, the when we showed the shortly in the slides before. So here, you know, this mutation from adenine to thymine, how it affected eventually and resulted in this functional protein. So this is kind of missense. Okay, so different types of mutations are ones that we can see. Okay, where either it's again one specific point mutation or a group of nucleotides at the same time. Nonsense, on the other hand, is when it's an early stop codon in the protein, so it's not a complete protein. So this also may result, you know, as compared to no effect, some effect, and you know, we stop there. So we don't get the full protein and full functionality. We visited also this slide before in one of the early modules, looked into variants and how there are different technologies that are able to look into different types of variants. In this case, we're looking at intra-read. So within the same read, we may face some types of, you know, specific types of mutations or changes like deletion, insertion, or inversion and translocation, so on and so forth, as compared to other types that could happen across two different reads. Okay, especially in the context of next generation sequencing. So let's have some examples and discuss these examples and see how we can explore that in the IGV viewer. So here is the reference genome. So we are using this reference genome as a guideline for us for what would be a normal case. Okay, so it is our reference. Okay, and if we, let me start with the one on top. So this is a reference genome. Everything is normal. Now we have a subject genome where is some deletion event. So how can we recognize this deletion event? Well, if we see the reference genome, and once we get the assembled subject genome, or in other words, we get the paired end sequences. Okay, so the expected insert size. So when looking at, you know, the paired end sequence, we are looking at one strand. Remember this. Okay. So I'm having A, C, G, T, T, A, right? I'm having some sequence here. And so imagine it's one, you know, paired end read. So I have A, C, G, T, T, and then T, A, T, T. It's the same strand. We're not looking at both strands, right? This is forward, this is reverse, and this is the insert area here, okay? So originally the insert area, if the if so if the reads are coming with a larger than expected insert size, like in this case, this is just a deletion event, okay? Something is missing here. We are expecting that there is some distance, okay? So this, refers to, of course, a deletion event. And it's easy to see this visualized, which we have explored in the previous part. You can see here, there is a gap, a very clear gap, okay? We're looking at the different reads, forward and reverse read through the normal or, the tumor cases. So this would suggest that there has been a deletion event in the case of that specific location. At the same time, we can look at the specific distance. We're going to recognize if that deletion is unique to the tumor case patient or the normal person and try to understand the differences. In another way, there might be an insertion event. So this is the original genome, reference genome. It has been subject to insertion. And now we can use next generation sequencing, because if the expected insert size is, or the reads is coming with a smaller than expected insert size, so this is the inferred insert size, okay? And now this is the expected insert size. We expect, okay, this, okay? So reads with a smaller than expected insert size suggest an insertion event, okay? So these reads here, as you would see in the subject genome, when we're trying to map them, okay, to the reference genome, we see that, they are having a small distance in between, okay, a small distance in between as compared to the expected area. In that case, we are saying, oh, we're recognizing an insertion event. There are other types of events that we may look at as well, like rearrangements between chromosomes. Okay, rearrangements between chromosomes. So in this case, this is the reference genome. We have chromosome 1 and chromosome 6. And then one part is flipped and switched with this other part. And so we look at the reference genome here. We're going to look at reference genome and looking at these, subject genome, trying to find how we can see or detect the chromosomal rearrangement. These chromosomal rearrangement can be, translocation, inversion, or large deletions. Different types of chromosomal rearrangements can take place. Okay, so in the case of translocation, where segments of two different chromosomes have been swapped between both sides, the forward read, let me show you this, okay? Yeah, the forward read might map to one chromosome while the reverse read maps to a different chromosome. So what do we see here? Well, let me explain this. We're looking in that sequence, the next generation sequence, you're looking at one strand. And so we're looking at the paired end of that one strand. Now what happens here? Usually they are part of the same chromosome. But now we see the reads are part or pointing. These paired end these paired end reads is being in different positions or places. So they are being mapped to a different chromosome. This is indicating a translocation. It is because the DNA fragment from which these reads come spans the breakpoint of the translocation. Spans the breakpoint of the translocation. As we can see here, with particular for the two chromosomes, so looking at chromosome 1 and chromosome 6, and we can see how mates are mapping to separate. And this in this screen here, it's a split screen view of the read alignment from the tumor sample and the matched normal sample, as we discussed earlier. And we have regions from chromosome 1 and 6. So in this example, alignments whose mate pairs are mapped to unexpected locations are color-coded by the chromosome of the mate. And other alignments are displayed in light gray. So we see these colored regions indicating, oh, there is a mate here. Okay, there is you know, paired end read. We're going forward and reverse, but instead of having them, did we see this in the previous slide? I think, yeah. Well, in the same slide even, we can see this, you know, it's, you see these directions here, right? And the opposite ones here, okay? So indicating, right, indicating that, you know, we have, well, sorry, so I guess, yeah, I guess that's, if we're looking at the normal case where we have the gray cases here, so the paired end would be these reads, okay. Not sure if it is correct to say this, okay. We're having those, okay, and as indicating, okay, maybe on the same side. Okay, so yeah, I'm just trying to think, okay, if we should see them on the same line, okay. Basically, we're doing these reads from the same strand and how they align on top of each other, okay, it's gonna guide us into, yeah, but yeah, it doesn't need to be in the same line, okay. So I guess it's fine to have that sketched as follows. So I have maybe one here, but in this case I have one here and one here, just for illustration purposes, or maybe this one here, okay, related to this. So these kind are, you know, showing us that, well, the mate pairs are in two different chromosomes. This indicates an issue, and if that kind of issue as well is what we can face in sequencing data as soft clips. We discussed before the idea as well of soft clips, which occurs when part of a sequencing read does not align to the reference genome. This is the reference genome. So part of that sequencing reads that we have is not lining up perfectly with the, you know, to the reference genome. And we call this unaligned portion as the soft clip by the alignment algorithm. Okay, so in other words, a section at either end does not align to the sequence. Okay. Okay. So what about inversion? You know, all of these examples are kind of illustrations for us in a way or another. This is what I say. I'm not certainly very expert with this kind of analysis, but let me highlight a couple of things. So When looking at these variants, we recognize that there are many types of mutations. Like in this case again, we have the reference genome, so there is an inversion. So this inversion, we have a change in the locations of these two sites. Now, can we detect that? This is the question here. Can we detect that using specifically next generation sequencing? This is what we are discussing here in this case. Can we detect this using next generation sequencing? And if we look at the reference genome, the natural part of that, again, we are looking here, and then these are two mates, meaning they are two paired end mates. So they're going in that way, opposite to each, sorry. So this is going in this direction here. Yeah. And this kind of alignment is indicating for us that it's the good case. Okay, so we're able to recognize that these are the parts of, this is the one paired end, this is another paired end, so things seems fine. However, when this is reversed, you see this is reversed now, this other one is reversed, and this one is reversed here. So we can detect this through the sequences, reading the sequences. And this is telling us there has been some inversion in the process. Okay, so in all of these previous examples, specifically here, okay, we're looking at the distance, for example, between the paired end sequences, okay, and this could be larger or smaller, indicating, deletion or insertion, and the reference expected area. And then, as well as, we're trying to see if they are distantly now in two different chromosomes, that's indicating something to us, translocation. Or we see now, oh, they are reversed. Some of them are reversed, okay? Some parts are reversed. These reads are reversed. So this is indicating for us, again, some changes. So to summarize, we can visualize this. This is the whole thing here. We can see this through an IGV viewer. And actually, this is where we're going to get to, is we can use some pipelines for software for variant calling. So whatever we have done so far, trying to visualize this as experts or building and training ourselves with this kind of analysis, what about, figuring out through running a pipeline or a software to get already the outcome of this whole process? In other words, let me run a program and let the program to detect the variants based on this analysis of sequences. Tell me if that's insertion or deletion or whatever. And so here we have the VCF format. Okay, the VCF the VCF format and the nomenclature. Okay, culture. So what's VCF? It is a standardized format to store information about genetic variants, right? We're interested in knowing these variants, single nucleotide polymorphism SNPs, or insertions, deletions, or other structural variations. So this file, as you can see here, typically contains metadata, a header, and data lines, or the body. The metadata is providing information about the file format. The header is giving description of the content and the data columns. Okay, so this is the metadata. Okay, and the header itself is giving descriptions also about the information that we have. So the data lines, as you can see here, each line contains information about a specific variant. Okay, about a specific variant, including its location, the chromosome position, the reference allele, okay, and so on and so forth, the quality score, and other data that can come, like genotype or other information. So these files are commonly used in genomic research, specifically in genome-wide association studies. Nice thing about IGV viewer that you can upload these files that you can get from one of the pipelines or the tools. Okay, so here is a question here: what is the allele frequency of this germline variant? Okay, so this is a treasure hunt question. Okay, I want you to try to think about it. What do we find here as variants, right? So what can you detect as some variants that we have? Okay, so you can see different colors here, right? For example, A, there's replaced with the G as compared to the reference. So what's the allele frequency in this kind of variant? Okay, so here we are looking at, we have 1, 2, 3, how many reads here? I guess you can go through it. Okay, so you need to count, you need to count the numbers and double check what you get here, because the total reads we have, I think 2, 4, 6, yeah, 22 reads. Okay, so 12 of those are having A and 10 are having G. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so these are the 10 cases here. Okay, okay, so this is indicating for us some changes that we can look at. So specifically at this case, we can see that given a reference of A that was altered with G, this is how we try to write down or represent this in the VCF format. We will have a quality score, okay, and the position of that and some other type of information. Let me give you here an example. If about the related nomenclature of different types of variants, because this came from the Human Genome Variation Society. There was this Human Genome Variation Society that tried to develop this. Otherwise, as a standardization, everyone can come, with their own way of how to format this kind of how you would write down a specific variant. So let's take this as an example here. This is NM_004006.2 and then colon C.76 and then A, then okay, arrow T. Now this describes a substitution where at the cDNA level, the nucleotide at position 76, okay, is changed from A to T. Okay, and the specified transcript, this is the label of the transcript, something we have seen before in the last module. Hopefully you recognize this also through doing the exercise and the activity. Okay, so this is how we can read this at the cDNA level. At that specific nucleotide, which is 76, it was shifted from A to T. And similarly, you have this example that you can refer to in the slide. Okay, so I'm gonna stop here and then continue with the last part about tumor suppression and oncogenes.